The narrative revolves around the varying effects of alcohol consumption, contingent on the amount consumed and the individual's unique circumstances. Essentially, the alcohol-induced impacts fluctuate from mild euphoria and decreased anxiety to severe intoxication and even unconsciousness. Should higher doses be involved, the potential of alcohol to penetrate almost every body cell is significant due to the permeability of cell membrane. Factors such as intake, amount, and context, including having food before consumption, dictate the degree of intoxication that can result. The presence of alcohol at elevated levels can elicit hangover effect, while extreme overconsumption can be fatal due to alcohol toxicity. Indirect deaths can be initiated by asphyxiation via induced vomiting. Sleep conditions might worsen with alcohol use, and during periods of abstinence, sleep irregularities can be a signal for future relapse. The universal definitions for alcohol servings or a moderate alcohol dose are inconsistent with the U.S. National Institute on Alcohol Abuse and Alcoholism, suggesting a standard of up to 2 drinks, 28 grams, for men and 14 grams. For women as moderate. Impacts of alcohol tend to vary as per the individual's blood alcohol concentration. Factors such as age, sex, and prior health conditions play a role. Even with the same alcohol intake, will differ among individuals, and consequently, the effects to individuals' alcohol tolerance levels also differ significantly. Curiously, there are findings implying slightly lesser chances of vehicular accidents with BAC levels between 0.1–0.4 than with 0.0.0. Known as the Grand Rapids effect, it suggested this might be a result of extra caution exhibited by drivers at lower BAC levels. However, this theory, like many others, remains speculative. Research indicates that individuals under the influence of alcohol maintain a higher degree of control over their behavior than previously believed, even though their capacity for evaluating the consequences of their actions is diminished. The environmental context is found to influence behavioral changes linked to intoxication. Areas related to planning and motion learning within the brain are, in fact, invigorated. An associated effect brought about by even low consumption of alcohol includes pronounced animation in verbal exchanges and physical gestures, enhanced metabolic rate and movement. Associated parts of the brain such as the nigrostriatal pathway, which in turn activate reward systems, are responsible for this increased vivaciousness. Alcohol's effect also extends to reducing the secretion of antidiuretic hormones that assist kidneys to absorb water effectively. Alcohol renders the osmoreceptors in the hypothalamus, responsible for transmitting information about osmotic pressure to the posterior pituitary, where the antidiuretic hormone is released. Unable to function correctly, consequently, the kidneys lose their capacity for optimal water absorption, triggering excess urine production and consequent dehydration. Excessive consumption of alcohol often results in acute intoxication, triggering immediate or long-term harm to health. Alcohol makes NMDA receptors unresponsive, consequently impacting brain functions. This is aggravated by alcohol stimulation of the gamma-aminobutyric acid (GABA) system, known to hinder brain activity and contribute to memory impairment. The GABA system, present within the hippocampus, is implicated in altering both stages of memory formation. Severe alcohol consumption can also lead to anterograde amnesia, known colloquially as blacking out. This refers to memory loss both during and after an episode of intoxication. Ataxia, or loss of full control of bodily movements manifesting as clumsiness and unstable gait, is another common symptom of alcohol intoxication. This is believed to be a direct result of alcohol's effect on the cerebellum. The Mellanby effect is another well-documented phenomenon, wherein the degree of behavioral impairment caused by alcohol is lesser when blood alcohol content, BAS, is decreasing as opposed to when it is rising, given the BAC levels are identical. This observation was solidified following a meta-analysis in 2017. The metabolism of alcohol relies primarily on 2 enzymes, alcohol dehydrogenase, AEA, and aldehyde dehydrogenase, ALDA, which can function differently based on gene mutations. Some genetic deviations could increase metabolic activity, particularly in the 8H genes, causing a buildup of acetaldehyde. Moreover, in the ALDA2 gene, a null ALA hinders the breakdown of acetaldehyde to acetate, leading to its accumulation. These gene variations can elucidate the disparities in alcohol metabolism among different racial groups. Ethnic groups like the Han Chinese, Japanese, and Africans appear to have a protective genetic variation. The presence of AdHonB2 in Han Chinese and Japanese and Japanese and Japanese and AdHonB3 in Africans lessens the risk of alcoholism. In contrast, the presence of the ALDA22 mutation in East Asians can spike acetaldehyde levels in the blood, causing symptoms such as facial flushing, nausea, and rapid heartbeat. Among others, these populations may feel the ill effects of alcohol earlier, preventing them from consuming it further. For Caucasians, the most widespread gene is AdHonB, which slows down the conversion of alcohol to acetaldehyde, making them more predisposed to alcohol misuse. Humans typically metabolize alcohol through NAD-dependent 8 enzymes, converting it to acetaldehyde, and successively metabolize acetaldehyde through NAD2-dependent ALD2 to produce acetic acid. Surprisingly, about 50 of East Asians exhibit a deficiency in metabolizing acetaldehyde. In-depth research in Japan discovered a single nucleotide polymorphism, SNP, variant allele of the ALDA2 gene, termed as ALDA22, that swaps an amino acid and renders the enzyme incapable of metabolizing acetaldehyde to acetic acid. Close to 57% of the Japanese population carries the normal allele, with 40 having the variant allele and 3 carrying 2 copies of the variant allele. Since ALDA2 works as a team, those with 1 or more variant allele contain inactivated ALDA2. These individuals metabolize alcohol normally but are unable to metabolize acetaldehyde efficiently, causing bodily reactions to alcohol ingestion, such as a temporary acetaldehyde buildup in blood and tissues, and facial flushing, among other symptoms. The story's central theme focuses on the genetic factors contributing to differing responses to alcohol consumption across various regional and ethnic populations. Around 35 of Brazil's native caboclo, 30 of Chinese, 20 of Koreans, 11 of Thailandese, 11 of Malaysians, 3 of native Indians, 3 of Hungarians, 3 of Hungarians, and 1 of Filipinos carry the GLA487 genotype. Either in a heterozygous or homozygous form, the numbers essentially fall to 0 among native Africans, essentially fall to 0 among Western European Caucasians, Turks, Australian Aborigines, Swedish Lapps, and Alaskan Eskimos. Among those who possess 0 or low levels of this genotype, over 5 commonly experience reactions to alcohol, characterized as ethanol reactors. These individuals may have other genetic abnormalities leading to acetaldehyde accumulation upon consuming alcohol, potentially leading to an allergic response. For instance, those with the normal AdHonB gene demonstrated a 16 incidence of self-reported ethanol-induced flushing reactions, as seen in the sampled Scandinavians residing in Copenhagen and Australians of European descent. Meanwhile, possessors of the AdHon ARG48, the SNP variant, showed a 23 incidence rate of such reactions. Their bodies metabolize ethanol so rapidly that acetaldehyde levels may surpass the metabolizing capacity of aldehyde. Even with these findings, experts propose that a significant percentage of people experiencing allergy, like symptoms from alcoholic beverages, particularly in populations with low GLU487 genotype occurrences, may stem from authentic allergic reactions to inherent or contaminating allergens, especially in wines, to a lesser extent beers. The story also explores alcohol's neurological effects. At low to moderate doses, alcohol acts as a stimulant, triggering dopamine release in the striatum and modulating GABA-A. It also has a significant impact on glutamate. Alcohol decreases glutamat's capability to bind with NMDA and acts antagonistically on the NMDA receptor, leading to possible memory loss. Chronic alcohol use can lead to an upregulation of NMDA receptors, which can cause severe symptoms such as delirium and hallucinations during alcohol withdrawal. Drinking a small amount of alcohol, such as 1 beer, can enhance overall sleep duration and reduce nocturnal awakenings. However, this beneficial impact on sleep wanes with the intake of moderate to high quantities of alcohol. Interestingly, one's familiarity with alcohol also contributes to how it affects their sleep. For instance, novices tend to feel calm after consuming alcohol, while seasoned drinkers often feel invigorated. Taking a closer look at the effects of alcohol on sleep patterns, it's noticeable that moderate drinking can initially improve sleep for insomniacs. Yet, consuming alcohol about an hour before bedtime, even when consumption is declining, interferes with the structure of sleep once the body metabolizes most of the alcohol. A rebound effect can provoke disturbances in sleep later at night. Moderate alcohol consumption can coincide with sleep disturbances in the latter half of an 8-hour sleep cycle. Moderate doses of alcohol can lead to rebound increases in rapid eye movement, REM sleep in stage 1, sleep in stage 1 sleep in the second half of the sleep cycle. Following initial suppression, slow-wave sleep, SLOOS can also spike quickly in the first half of the sleep cycle. These modifications in REM sleep and SLOOS after moderate alcohol consumption are related to the reduction in glutamatergic activity by adenosine in the central nervous system. However, tolerance to changes in sleep can develop within 3 days of consuming alcohol before sleep. Notably, alcohol can also influence balance because it alters the density of the endolymph, a fluid in the semicircular canals in the ear, resulting in exaggerated body movements and potentially causing the spins feeling. Additionally, consuming alcohol with food enhances and extends post-meal fat levels in the blood. This occurs regardless of the AJ-shaped correlation between alcohol intake and fat levels in the blood, illustrating lower fasting triglyceride concentration among moderate daily drinkers compared to teetotalers and heavy drinkers. A 2015 literary survey unveiled intriguing links between alcohol intake and pain management. Notably, the research shows that moderate usage of alcohol could lead to beneficial effects in relieving pain. However, it's not all positive since the study further noticed a curvilinear correlation. Moving towards heavy alcohol consumption resulted in adverse impacts concerning pain-related outcomes.